Okay, thank you so much for this kind invitation. First of all, happy Mother's Day to everyone, and don't forget to say something to your mother today, please. I guess you would prefer to have Mother's Day on another stage, but today is my turn, only 40 minutes. So thank you for that. And also thank you, David, thank you, Cherry, for the kind invitation. And I would say, thank you to everyone, because honestly, I built my career in extracellular vesicles. I started to do that in 2008, and now it went really pretty good for me, I have to say. So whoever is PhD here, just telling you when I started, I knew nothing about EVs. Now it became like that. So thank you for this invitation. Today I would like to share basically, I put this title like Beyond Borders, because mostly I work in metastasis, and we've been physically thinking that the edge of the tumor is the primary tumor, but I became aware that EVs could be a new border for the tumor. Basically, they are shed out. They could be considered at the front barrier of the tumor. So today I'm going to discuss this idea with you guys. I am giving you not only what we did, but also a critical view of what we are using, or what is the concept of extracellular vesicle in pre-metastatic niche formation, right? So starting with that, I would like to start with an overview of the metastatic process, and for you to consider that metastasis is just evolution of a tumor cell. You have the primary tumor. Those tumor cells, they really need to learn how to survive in circulation. They could be actually interacting with platelets in the circulation. They can go to distal organ. They will need to learn how to survive over there, and they will survive and succeed. But this is really inefficient. Only 0.1% of the cells coming out from the tumor will go and land in those metastatic organs. And figure out in the field like that, and in this crowd, you are probably missing something. And this is what I did when I started. When I started this, I was wondering if extracellular vesicle could be playing a role in that. And as you see here, we were missing a piece of the puzzle, right? Extracellular vesicle can be secreted by any cell type, not just by tumor cell, but any cell type could be contributing to that. And we were trying to understand how. So today I'm going to give you an overview of the role in pre-metastatic information, but also I wanted to give an overview of the role during tumor progression. So I would start with key role of extracellular vesicles in the primary tumor growth. I'm not going to talk much, but just some examples. Like you probably know Janus Rack, right? He was one probably of the first, like Shanda Breckfield and all this, telling us that, okay, guys, and this is what it catches my attention when I was over there, that material in an extracellular vesicle could be transferred to another cell, can be, I call it corrupted, a corruption mechanism of other cells. So there is a horizontal transfer or influence of other cells within the primary tumor, but not only between tumor cells, but also tumor cells with cells in the microenvironment. So the idea was that, and we know now that not only tumor cells can do that. This is just an example. We have CAFs, for example, we have endothelial cells. It's a horizontal transfer of material. So that was fascinating for me. I have to say that when I started this, okay, I like this field, I went in Gothenburg the first time. I like the people dancing over there. So, why don't you build my career on extracellular vesicles? And we now know more, right? We now know that in the primary tumor, there is also influence in epithelial tumor mesenchymal transition and also in immunosuppression. As I said, these are only examples, 4 fields I talk. But one of the things I learned when I was in the US, I did my postdoc with David Leiden, you know him, and they settled this concept of the pre-metastatic niche formation. And today I'm going to tell you a little bit how it looked for me. I didn't know what to do in my postdoc. I went to another lab. I didn't get it, and I started, David, would you like to meet with me? Yes, he said, yeah. He came to me and said, Hector, we are working on this, and this concept of the pre-metastatic niche formation is just, while a primary tumor is growing, it's secreting factors. At that time, only soluble factors were actually new that were contributing to that, and they are somehow priming new soil for metastasis. This is the seed and the soil I'm going to go over later on. Somehow, a tumor cell is able to metastasize, but if you prepare this soil, it's going to metastasize much better. This is the concept, right? So David built this paper in 2005. So for me, it was really interesting, this topic. This is what we knew at the time, and when I got into the lab, soluble factors, TNF-alpha, TGF-beta can be secreted. They promote several hallmarks of the pre-metastatic niche, I will tell you later. They promote the expression of pro-inflammatory proteins, several extracellular matrix proteins. They define in this paper that hematopoietic progenitor cells can come over and develop this pre-metastatic niche before, and this is the key point, before any tumor cell is there. So anything was happening before tumor cells were there. So for me, it was fascinating. And also I remember perfectly that day, David came out, okay, Hector, we have slides from lungs, mice that have a primary tumor and cherry positive, just in tumor cells, and we see some dots in the lungs. What is that? And he came to me like that. Would you like to do a postdoc in this? I knew nothing, as I said, nothing. I said, okay, just take it. And then I took it. And this is what we wanted, right? Is pre-metastatic niche formation, the extracellular vesicles could play a role in that. And before going into the details, I wanted to share you this hypothesis that was settled by Stephen Paget, this the seed and the soil. You probably are aware, but he basically postulated that metastasis is not a random process. It will happen in a specific organ sometimes, and this will need the right microenvironment to settle. Not all the cells are able to metastasize on all the organs, you know probably. Every tumor type have their favorite metastatic organ. And we wanted to analyze this, and I mean, we know biologically it could happen, not strictly we know everything, but we know how it could happen in a specific tumor type, but we knew nothing about extracellular vesicles. Later on, we have Fiedler, Isaiah Fiedler, who experimentally demonstrated this hypothesis. And I have to say that Joan Massaguer, who is also an important investigator, is now in cancer research in Sloan Kettering, is the director of that. They settled this idea of organotropism, so those cells are able to metastasize in specific organs. He basically settled this at the intrinsic level. You know, intrinsically, the tumor cells, they get some information that allow them to metastasize in other organs. We are trying to understand this. It's not that I am saying that we know everything. No, no. We are trying to understand why and which factors are influenced. But our role, at least David's and mine, was, okay, are we going to look at the extracellular vesicle and see what is going on, right? But before, I prepared this as a story. I hope you pardon me, but I'm not going to tell you exactly my paper I published, but I want to give you an overview of the field. So that was the first question that we had, okay, do EVs reach distal organ? That was my main question. Do they promote changes in the pre-metastatic niche? And if so, is there any influence in the metastatic niche formation, right? So let's start with the first one. And I wanted to put some question first, you know. Sometimes we receive questions from reviewers, but this is more a critical angle from my talk, right? Are we using the models that could represent physiological metastasis? This is one of the questions that we mostly are. Actually, we know that results mostly depend on the methods that we use. Sometimes we have variability, not just because you use one model, you use one isolation technique, but also one labeling technique. And these are things that we really need to think about it. Or at least, what I tell my people is just to be aware about it. You have to know what did you use, why, and if you are able to have the limitation of your material. That's it. You know, at least to define it, right? I'm not talking much about liquid biopsy, only at the end, but there are pros and cons in liquid biopsy. Are we able to use extracellular vesicles in diagnosis, in prognosis? Or are we aware that tumor-derived EVs in circulation, there are just a minority of them? So these are things just I would like you to think, or you know, a reflexive angle today. Also on this, what I tell my people is, okay, I am sure that we are not using the protocol for isolation, the best protocol. I'm using one of them. So in the community, we really need to do so. We really need to specify what we are doing so you are able to think about it and, you know, how we did it and how we look into the, you know, biodistribution, specific ability. But if we are specific, at least we can define what we did. So this is what I'm going to tell you today. This is the answer to the question that I'm going to talk today about this. You probably went through this every submission, but also every day in your lab, right? Is the dose we use physiological? Okay, I don't know how many times they asked me about that, but I can tell you that in my case, what we are doing most of the time is injecting the dose that is equal to 1 million cells. And this is because I talk about Joan Massaguet, right? And in cancer field, you normally inject in the flank 1 million cells. Is this physiological? That was a big answer, a question I have. Is this physiological? No, it's a model, so we are trying to model the disease. This is what I wanted to tell you today. We are modeling the pre-metastatic transformation. I'm sure we can discuss forever on that, but I would like to say what I'm doing here. Are you sure they are pure? We do these 3 markers. Do you use the protocol? We use ultracentrifugation in this application. Are they representing the heterogeneity? Well, we are doing analysis of cell line. This is what we are aware, and cell line derived from, EVs derived from those cell line. They reach distal organ, and actually, the mechanism I will show you now, and the idea is like, did you rule out the role of secretive factor? Depending, sometimes, if you use ultra, if you use other techniques, not. And is the effect, your favorite gene? In this case, you will apply to your specific modality. The idea is, I hope you can be inspired by these ideas that I can share. So this is how it all started. We wanted to analyze the biodistribution, and you have to be aware that depending in the area that you inject EVs, it's going to be totally different. If you inject intravenous, they will likely go into the lung and the liver. If you inject intracardially, you will get it into the brain and the bone. If you inject in the foot pad, you will get it into the lymph nodes. And this is what happened. And actually, these are equal models. So one thing to have in mind, that irrespectively how you label EVs, if you inject in different areas, it will go to different organs. In that sense, you have to understand the biology behind your tumor in order to understand the pre-metastatic information. This is the key point we wanted. Which are the hallmarks of the pre-metastatic information? With David, we published it. One of them is vascular disruption. This is first hallmark you get. Then extracellular matrix remodeling, and inflammation. So these 3 key points, this is what we were chasing in our material, right? The first one is vascular leakiness. If you inject mice in the tail vein, these are lungs, you will get vascular leakiness. This is dextran you inject after injection of EVs. If the dextran leak out, like is in here, you have leakiness. So we were basically sure that this was happening at least in the lungs. But if you inject in the foot pad, like looking at another organ, then in the lymph nodes, you will promote leakiness. So this is what I wanted to tell you today. So if you look later in the brain, for example, you will see leaking out in the brain, but it was injecting in the heart. So depending on the model that you would like to change, you will have the physiological effect, right? So after this time, I think that pretty aware that during the pre-metastatic information, we are actually looking for different mechanisms. One is vascular leakiness, the another is extracellular matrix remodeling, and then we have the bone marrow cell recruitment. This is what we got, and this is what we are having normally in our field. After that, you will have the pre-metastatic information. Then, I would like to share you today, but this is all published work, but those works that we published time ago, this is my postdoc work, right? When you were looking into that, EVs are able to reach distal organs, in that case lungs. We promote the pre-metastatic information, vascular permeability, extracellular matrix remodeling all the time, and also another observation that we have a systemic effect. So we are not only having a local effect in those lungs, but we are having a systemic effect in the bone marrow. And for me, honestly, was, what is that? So we are having mobilization of bone marrow cells. In this case, we defined that c-Met, the HGH effector, was influencing the mobilization of bone marrow-derived cells. So thinking about it, okay, what is this strategy? The tumor is sending something to the lung, to the bone marrow. They are promoting mobilization. Honestly, you have to see that in a physiological angle as well. So physiologically, the pre-metastatic information is just a damage that is happening in the lungs, and the bone marrow cells are likely going physiologically to repair that damage. So we have to understand that the pre-metastatic NIF formation is the pathological reflection of a physiological aspect, right? Just for you to understand what I'm thinking about. So after this paper, of course, we became aware that many other organs could be into that. Lymph node metastasis we published later on this work, but before that, with David, actually, we defined together with Ayuko, Bruno, Tanlon probably is right here, so we defined this role of extracellular vesicles in metastatic organotropism. And that was honestly one of the, smart ideas that David had. Okay, if we are chasing this organotropism, can we actually have organ-tropic models? And this is Joan Massaguet's model. You have lung-tropic, liver-tropic, and we would like to define the cargo of extracellular vesicles and if they can reach those distal organs. So after many years of work, we came with this. We knew that a specific extracellular matrix in those organs, such in the lungs, is slowing down EVs in this, and these EVs were carrying a specific integrins, like alpha-6 beta, oh, sorry, I cannot see, beta-5, sorry. I really need my glasses. Sorry, alpha-6 beta-4 and alpha-6 beta-1. And we knew that fibronectin was coming out in the liver, so alpha-B beta-5 is coming in there. So that was something we knew. But not only that, they slow down. So I see this as a dynamic mechanism. Honestly, this is like a population. Imagine there's a lot of population, and you guys are exosome coming from the tumor. Where are you going? You are coming to Australia because in Australia is ISEV. So ISEV is going to be fibronectin and laminin, and we are all exosomes, we go there. So that's it. It's a dynamic movement. That's the inspiration. So we found out that they promote the expression of specific proteins in those which are pro-inflammatory proteins, and then bone marrow cell will come and promote the pre-metastatic information. And after that, we realized when I moved back to Spain and that there was missing a piece of the puzzle. You probably know the metastatic spread. There is a way of metastatic spread which is hematogenous, you go to distal organ, but there is another way also that is locally, that is lymph node metastasis. It's mostly well known in the field, like in melanoma, in breast cancer, you will have that. So I wonder whether the EVs could be influencing the pre-metastatic niche. There were several works already telling us, okay, EVs could do that. There were several other authors already proposing that, but we wanted to look this much more in detail. So in that settle, we settled the system. We are going to use organ-tropic models, like models derived from lymph node metastasis, and see what they do in the lymph node, right? And we came up with this story that was published in 2021, where we observed that specific proteins like NGF receptor is also known as P75, whoever is working with that, sorry about that, because it's the most well-known name, is P75NTR. We saw that this protein is secreted in extracellular vesicles. It is horizontally transferred to lymphatic endothelial cells. Lymphatic endothelial cells start to express a nerve growth factor receptor, and this is triggering ICAM expression, activation, and lymph node pre-metastatic niche formation. So for us, it was really, really a burst of energy because we saw that lung metastasis, then lymph node metastasis, so it could play a role overall in the pre-metastatic niche formation. How? We settled this system that probably you know about the education system. And we came out with this idea, okay, actually, when I was there, how can I mimic the effect of EVs in vivo, right? You may agree or not, but this is what we did. We isolated EVs and then injected mice. So you can consider this as a treatment. It is a treatment, but I call it education because EVs are so complex that it's not just TGF-beta. We are calling like a massive amount load of protein, microRNA, so this is what I call it education. What I'm doing today is education. Yeah, I'm giving you a speech. Whoever is giving me a speech is educating me. So constantly, if you come to hear me every other day during the next 3 weeks, you probably will learn something about my lab. So this is the concept. So we try to do this. We put in the flank of the mice a tumor, and we educate it just in the footpath in order to promote lymph node metastasis. If you inject in the footpath, the material get to the popliteal lymph node. So we were aiming to promote pre-metastatic information in the lymph nodes and injecting the tumor in the flank. And after that, what we observed is that, okay, somehow the educated group was developing more metastasis. And this is what you use to long-term study. So this is what we call ongoing education. You inject EVs along the time course. But we have also education to detect the pre-metastatic information. And this is when you educate before only. And this is normally what we did, most of the time. We educated mice, and then we inject primary tumor and see if this happened. Hopefully, we could do this with many models, but at this time we were looking for lymph node metastasis. We educated food path, and then we injected tumor cells, and then we could see those cells who were overexpressing NGFR, they were going more into the lymph node and actually going further, metastasizing. And we tried to demonstrate that the other way around, educating with knockout models. This is the tricks we use in the field is, okay, let's remove NGFR in the cells, so we use NGFR knockout EVs. So we were using the two models, NGFR expressing or knockout. And in this case, what we observed that we can slow down metastasis. And this is how we engineer the models in our system. We knock out the gene that you like in your tumor cells, then you use EVs, either knockout or overexpressing, to model it. And again, I would like to emphasize this, we are trying to model. Because, of course, you can tell me, Hector, NGFR is not just the protein, it's in the EVs. Totally agree. As I said, we have pros and the cons. So we have to assume that we are not perfect, and we are trying to model a model system, right? So in that sense, after those years, we come out with this story, really nice story with David. Of course, I had to acknowledge him and all my co-workers in David Lyle's lab, because we came out with different stories that we having set in this pre-metastatic niche in the lungs with CMET, that was my work. Then we have a UCOS paper together with Tan Long and Bruno about the organotropism. Bruno published this about the pre-metastatic information in the liver, right? And then Gonzalo came out with this idea in the brain. And after that, when I came back to Spain, we defined this story. So as you see, honestly, I'm really, I think we round the circle about the pre-metastatic information. But it wouldn't be fair to say that we did that by ourselves, but because there is much people, of course, in the field, and that's, probably you are in the crowd. Thank you so much, because I think we all together contribute with many other. Maybe your ideas are in the slide, maybe are not. I apologize because I couldn't put all them, but I selected some works along the road, which they contributed, for example, to the, ifs the other day about the macrophages or the recruitment of macrophages into the pre-metastatic niche, some related to the metabolism, some related to the angiogenesis, CD44, many others contributed to the generation of pre-metastatic niche in the lung. But not only that, of course, we know several roles. Other microRNAs are involved in the pre-metastatic niche in the liver, and some other, they also contributed to the pre-metastatic niche in the brain. And finally, the work that we publish is not by itself, but it's also. So I have to say that this came as a community work that I acknowledge, of course, that everyone could contribute that to generate this system and this model, right? So in that sense, one of the things is how it works, and the other ways is to see, okay, can we target that? And this is what we are working right now in order to target this mechanism, right? So you probably know this, how we can at least see this in a therapeutic way of view is understanding the physiological mechanisms that are involved behind. And this is what we do here. We know that angiogenesis is involved, bone marrow cell recruitment, vascular remodeling, lymph angiogenesis. So what we realize is that we are able to block specific receptors, at least ours. I can tell you about my work. We reduce this pre-metastatic information, and we kind of revert the effect. I'm not going to say we cure metastasis. We didn't cure metastasis. We slow down metastasis. So of course, our aim would be to combine this with other techniques in order to find definitely cure. Actually, yesterday we were discussing maybe immunotherapy, the combination with some other ideas. So I believe these are key questions that we need to face as a community. Before entering the next step or next ideas in my talk, I would like to think, I put some slides for, just think about together with you guys, is this concept to have in mind that EV cargo evolves as the tumor does. So of course, the tumor is in a constant evolution, as you see in the slide, and they are also the extracellular vesicles. So of course, extracellular vesicles from the primary tumor, the first one are not the same. Sorry, I lost my track. So the first ones are not the same that this extracellular vesicle. So, for example, the model we use for organotropism, that were these cells that are radically different than these cells, that evolved. So this is one thing we have to think about it. And the modeling system that we use is, if we would like to demonstrate organotropism, we need to go here. But maybe you would like to demonstrate the role in the primary niche, you need to go here. And this is just a topic I would like, you probably are aware, but I wanted to, raise this point. Also, and it was actually a question for me when I started, you probably know, Gracia Raposo, of course, and their work, and together with Silvergeld, that they demonstrated time ago that the cargo in EVs, these are dendritic cell-derived EVs, were immunogenic, and actually, in this paper, EVs from the dendritic cells, they were slowing down. And honestly, when I was starting my postdoc, I was not expecting that EVs from melanoma cells were actually inducing the premetastatic information. My hypothesis was this. I mean, maybe they could be used for vaccination, right? We are thinking about it. I was wrong. And this is how we did it, just because based on the on works that we read, this is something that we can learn. And my hypothesis was that, but this is what we are right now. We know that depending on the contributions of either tumor EVs or immune cells, in this case, antigen-presenting cells, you are going to have a different outcome. And if you put that in an in vivo context, it's going to be really, really messy. I don't want to overwhelm you, but you are aware of that. And the last slide in here, thinking about it, is this. We learned a lot. We are coming from dirty bags to the gold chest, right? And this is a critical angle also. Sometimes you flip the coin, right? Or we say in Spain that you change the glass, the color of the glass. Maybe you say, how do you say in English? Well, you just change the angle, right? So you see in one angle, you see that oncogenic proteins, harmful material is in this, toxic content, small proteolyzed things in the EVs. These are just, Hector, just stop doing EVs because these are junk. Okay, just flip the coin and look at this. Okay, maybe you can look for novel biomarker, you can avoid intrinsic damage, spelling out the EVs. Then you can find horizontal transfer of material, receptor-independent uptake, a specific biodistribution. And this is what we came in the field, right? This is what we changed. I changed that. I try to motivate my people. Don't look into that. Just think about novelty, about new angles. Maybe you're right, maybe I'm wrong. But this is what we did, or at least what is still motivating me, probably you guys, right? So this is one of the things I would like to encourage young fellows. I saw that we are encouraging young fellows into the crowd, so keep going. I knew nothing about EVs when I started. Now I'm here. You probably know about EVs, but you don't know what you know in 20 years from now. So, keep it going. So that's my main message, right? And in the last part of my talk, I would like to shift a little bit into the liquid biopsy. I am sharing unpublished data that we are getting in the lab for the last 3 years. And it's based on this concept of the use of novel markers in extracellular vesicle in liquid biopsy, right? This is really general content. You know that depending on the biomarker, you can either apply for diagnosis or prognosis. But one of the things I would like to highlight today is one of the questions I have all the time is, Hector, what should I do? What should I look for, right? You tell me, right? I don't know, But we decided honestly to go for that specific content that is in this slide, but it's not strictly. But we decided to go for small proteins that are on the EV, so are secreting out microprotein. They are well known as microprotein. And I'm going to explain what our microproteins are, what we are doing right now. Really briefly, you know that in the genome we have this open reading frame. They are small open reading frames, all localized in our genome. And I would say that this is like when we have RNAs, there were somehow, you probably remember, someday you see a paper, okay, there are microRNAs. What is this? So this is the same, it's the same equality. So microproteins are codified by a small open region along our genome. You can identify them in silico, you just profile it, but you don't know if they are expressed or not. They are codified, you have your mRNA, and these microproteins will be all along this there will strictly a machinery that will translate them or not. So we are trying to understand if we can see the microproteome, it is expressed or not in the EVs, and it is secreted. So the characteristics of the microproteins, they are below 100 amino acids, they are conserved evolutionary, and so far, some of them have been characterized, and some of them not. In other words, we know nothing about them. At least I know nothing. We probably, if there is someone who knows nothing, just tell me about it later on. So the idea is we became aware of this. We are starting to analyze the microproteome in EVs. And how we are doing that? We are doing several angles, but I'm going to tell you this. What we do is explant from the primary tumor. This is focused in pancreatic cancer. One way to analyze is to go to cell lines, but we decided to mimic more physiological secretion of EVs. We are getting explant from the OR, so the surgeon is getting us this piece of the tumor. We are getting the supernatant, and from this, of course, you can do the typical proteomics that we all do, but you can use a specific database for microprotein and identify. So the same supernatant can be profiled for proteins and microproteins. This is just the big thing. So we decided to go for microprotein because we had evidences that they could be pro-metastatic. So, and this is how it all started. And I say that it all started because you will see that it's not that easy. We've been working with 3 years, and we only came out with 2 that could be potentially that. So, but it's a funny story for a PI, not for the fellows. Sorry about that. But honestly, we tried to get that. The identification of these microproteomes, at least in the highly metastatic models, came out with 599 candidates all over the genome that could be secreted into that. And of course, when you come out with this, it's okay, we have to narrow down a little bit more. Okay, so how can we do that? I just put here what is the representation and where are codified. So now, most of them are in the 3' OTR, some of them are in pseudogene areas along the genome. What is novel about it is we could identify the origin and the region, but we still don't know if they are, because this is all in silico, we don't know if they are actually expressed as a protein. So the next work is to say, okay, we identify them, then we are going to see if this is true. And this is what I'm doing right now. Asking myself, is this true? Can we trust it? So the idea is like that. From those 600 in silico candidate, we established several, characteristics. They need to be enriched between tumor and non-tumor. They have to be conserved along the evolution. And then we should have some evidence on functionality, so you can see the structure and see there are domains. We came out with 23, so it's not bad, right? From 600 to 23. And then we started to clone them. So you take this theoretical micropeptide, you clone it. Remember, these are really unknown proteins, so you don't have antibody. You cannot go to Atcan, Santa Cruz, whatever, to get it. So you just have to do normally fusion with a tag like HA and see if you can express it and it's go. So we cloned 23, we expressed them, we saw if they went to extracellular vesicle or they didn't, if they could play a role. And we end up with 2, and this is what we got. Which is fine, so far so good, right? So we have 2 candidates named 9 and 13. I apologize, but this is the original name we gave it. It's nothing now, we can call it whatever. But the thing is, those are, as you see here, expressed exogenously, so we target them, and they are in the small extracellular vesicle. There is also some in the large, but as I say, we are starting this project. We believe this could be a potential secreted microprotein. We are trying now to see the endogenous expression of this microprotein. And as I told you, we don't have, like antibody, we don't have tools to do that, but we are aiming to do it. We are thinking, how can we analyze the endogenous expression? The exogenous expression is like that. You have a model, you overexpress it, then you see that is secreted in the extracellular vesicles. It is, you find these markers, and you basically say, okay, my molecule is secreted on extracellular vesicles. It could be done, and that's it. But besides that, we wanted to look about the functional effect, right? So in that sense, what we did is we overexpressed it and we saw what is the effect on the primary tumor and in metastasis. In primary tumor formation, in one model of pancreatic cancer, you see that one of the candidates, the 13th one, was getting the promotion of primary tumor growth. Again, we don't know how. It is promoting primary tumor growth. And also, we could observe that they promote liver metastasis. So basically, the concept is that we have a novel cargo, potential novel cargo, that could be going into the extracellular vesicles, the microprotein. I have to say that this is one probably a minimal piece of the puzzle. This is pancreatic cancer models coming from human tumors, but there are many other tumor types we haven't analyzed. So depending on the angle you would like to explore, it could be radically different. Of course, your idea here is to compare healthy tissue with tumor tissue, then you express it. And right now, what we are building upon this is a novel story. We hopefully find that. And the model we are chasing here is this. We postulate that primary tumor cells can secrete EVs. The cargo is really complex. I added a little bit more complexity into my models now with these microproteins that we should have in mind, as you guys have in mind microRNA. So one of the things when I started this is, okay, if microRNAs are somehow hyper-secreted as compared with RNAs into EVs, why don't microprotein could be the same model? So they are a small protein, they could be. The other thing is if they can play a functional role, right? We know that at least the candidate 13 is secreted in EVs, the 9 as well, but the candidate 13 is promoted. We have some data supported it's promoted not only in cell proliferation, also in cell invasion, and not only intrinsically, as we saw, but also extrinsically, so we can actually treat tumor cells or calves with extracellular vesicles, and we can promote their migration, and we hopefully think that this could happen in the future. The idea is to see whether they can play a role in the pre-metastatic initial as well. This is my field, but also one of the things we are looking at together with Klaus Pantel, Matthias Laura, and Nuria Malatzis, we are involved in this Mission Cancer project, that the idea is not just to see of course, we always chase the biology, right? But in this project, we also wanted to see a potential use as a biomarker. Yesterday, I was talking to David, right? We have this plasma from patients. Can we identify this protein as a potential surrogate markers of early primary tumor? And this is the project that we are having. This pancake project is trying to identify novel microproteins in the circulation in order to be potential biomarkers of early pancreatic cancer. This is a totally different angle that is not related to candidate 13, but these are the two angles we are exploring in the laboratory. If you are interested, you know where I am. The second to last slide is this one. I would like to present, HEIVEX, Spanish gang of EVs. Unfortunately, not much of them are here. Carmen is here over there, but in the board, what really enthusiastic is about science in extracellular vesicles. You know us probably. We are like a family in Spain. We have a good weather, but also good character, and we really get along together. We have a symposium in Mallorca, which is like here, like Australia, but with sun and good weather around in October. So it's a smaller island, no? But anyway, you can have fun. You want to stay there, of course, you are welcome. And the idea is to develop this meeting in October 23 to 25th. And the last slide, I stick to the time like perfectly. So, because I'm done, this is my people. Yesterday I put it here. We are surrounded by extracellular vesicles in my laboratory. I would like to acknowledge mostly Susanna, who is the first author in this paper of the Nature Cancer paper in NGFR. But of course, I have to say that all the persons in my lab are contributing. We don't say, you have your project and that's it. No, we have a collaborative laboratory. So all of them, I have to acknowledge their contribution to our laboratory. I would like to thank especially David, of course, because he gave me the opportunity to develop my career in extracellular vesicles, together with Jackie Bromberg, which was also a Sloan Kettering. Babak Menara contributed as well. So that was my US period. I really acknowledge their contribution to my career. I am big, like I am because of them as well. So I would like to acknowledge their contribution. Collaborators that we are ongoing here in the pancreatic cancer project. We have the project that I presented yesterday in immunity. So the idea is, I apologize if everyone is not here because we don't have, I had to put much sticker here. I decided to put all the stickers I have around my career. So I don't have, I mean, I have money, but I don't have that much money. It's just, this is a timeline story. So I would like to thank, of course, the funding sources, the people involved in the work, and you guys for your attention and work as a community. So thank you so much.